from this, what's called X-ray crystallography, from looking at the structure of virus crystals. The other thing that viruses, you could do with viruses, is that they form a kind of solution, a suspension, if you like, but it's a sort of solution called a colloidal solution, which is another thing they have in common with non-living things. So you can't really answer this question. You can just say a bit about it. You can't really say what the answer is. How do they multiply? Well, this just shows you it in a little bit more detail. The virus attaches to the surface of a cell. This virus has RNA, some have DNA, but it can have either RNA or DNA. And using this RNA, it forms proteins by the normal process that occurs in a cell. These proteins then assemble into virus particles, and they are released from the cell. And of course, this whole process is very disruptive to the cell, and that's one of the reasons viruses cause disease, because this quite often kills the cell. Can have other effects, which I'll come on to later on. So some examples of viruses. As I say, viruses have nucleic acid. This can be DNA, and all other living things have DNA as their genetic material. We have DNA as our genetic material inside our cells, and that contains the genes which make us function. And the way viruses function is also to have genes. And so they can have this in the genes in DNA, but viruses can also have RNA as their genetic material, and in this way they're unique. Viruses having RNA are quite different from all other living things, if that's what they are, in having RNA as their prime genetic material. So there are these two different kinds of viruses. This isn't the end of the story. In fact, there's 7 different kinds of viral nucleic acid, 2 groups of DNA viruses that are different in the way that their structure and function of the nucleic acid, and 5 groups of RNA viruses. It's called the Baltimore scheme. I don't really have time to go into it now, but I won't, but just make this difference that you can divide viruses into these 2 big groups, but RNA viruses in particular differ in the kind of RNA that they've got. Coronavirus is an RNA virus. It is what's called a positive RNA virus. So this is the kind of RNA it has. It's called positive stranded RNA. The reason it's called positive stranded is because the RNA actually codes for protein. So it can be translated directly into protein. Take another virus like measles or influenza. These have negative stranded RNA. They can't make protein directly. They have to be transcribed. And there are other kinds of RNA as well, but it doesn't really matter too much. How are they transmitted? Well, by far the most common way that viruses are transmitted, and there's very large numbers of viruses transmitted in this way, is by aerosol. And I don't think I've put it on this list, but coronavirus is transmitted, we think anyway, mostly in this way. So this is the formation of droplets when you breathe. And when you're just breathing, you're all the time breathing out these small droplets of moisture, and these could contain virus. Of course, if you cough or sneeze, you greatly magnify the number of droplets that you produce. And somebody hasn't muted themselves. And so yes, I've lost the track. Coronavirus is transmitted in this way, along with things like flu virus and common cold virus. The virus might multiply in the lungs, but other viruses are transmitted in this way, but they go on to infect other parts of the body, like measles, for example. Another way that they can be transmitted is in food and water. Poliovirus is transmitted in this way. And there are other viruses that cause diarrhea that are transmitted in this way. Another way that they can be transmitted, and we think, but we don't know that coronavirus might be transmitted in this way, is on objects that you touch. These have a technical term, they're called fomites. An Ebola virus is only transmitted in this way. So, as you can see, some viruses might be transmitted in more than one way, but usually there's a main mechanism of transmission. Then there's venereal transmission, and we all know about human immunodeficiency virus that causes AIDS, transmitted venereally. Herpes virus is another one that's transmitted venereally. Vectors can transmit viruses, things like mosquitoes and ticks. Yellow fever, for example, is transmitted by mosquito. And then what I've called artificial transmission. That's by things like blood products and blood transfusions. And we all know that HIV is transmitted that way, so is hepatitis C, and there are a number of others. So that's the way they're transmitted. What about their structure? Well, they have to have a stable structure, and this really means that they have to have some kind of symmetry. So there are two main kinds of virus symmetry. First one is what you might call helical, it's called helical symmetry. This is a coiled spring, and the nucleic acid would be wound along the inside of this spring, and these structures around the nucleic acid are composed of protein that's made by the virus. So the whole thing makes something called a capsid. Capsid is just a Latin word for shell, actually. So this is what the outside of the virus, of some viruses, is. The other kind of symmetry is called polyhedral symmetry, and this consists of equilateral triangles. Usually it's what's called an icosahedron, which is a very stable, thermodynamically stable structure, consisting of 20 sides, each one of which is an equilateral triangle. So here's a virus that's got DNA, which has this kind of structure. But for many viruses, there is another layer around this kind of symmetrical structure. And this is derived from the host cell membrane when the virus is released from the cell. So it's an extra layer, and this is called an envelope. And it forms a layer over the helical capsid, which you see here. So it's an extra layer. And often there are these spikes in the membrane, and their function is to attach to a new cell. These are viral proteins. So coronavirus is of this enveloped type, and we're lucky there because it means that the virus is easily inactivated. This is, as I said, this envelope is derived from the host cell membrane. This means it has fat in it, it has lipid in it. And lipid, of course, is soluble in soap and detergent. So if you treat coronavirus with soap or detergent, you dissolve away the envelope and you inactivate the virus. So that's very useful. and is an easy way to disinfect things that have been in contact with the virus, particularly hands. If you wash your hands with soap and water, you get rid of the virus, theoretically anyway. So many other viruses like norovirus don't have an envelope, and they're much more difficult to inactivate. So we're lucky there. A few viruses have a combination of these two kinds of symmetry. I won't go into that. That's a bacteriophage which has the combination. Now, I said that you can't see viruses in a light microscope because they're too small. The reason you can't see them is because the wavelength of light is too long to be able to visualize a virus. It's a physical impossibility. But you can use electrons. Electrons have a much shorter wavelength than light, and you use an electron microscope, and here's an example of an electron, or a photograph of an electron microscope. It's like a cathode ray tube, if you like. You remember the old television screens that used to emit electrons from one end and they hit a fluorescent screen at the other, and that's how you got your television picture. They're not made that way anymore, of course, but an electron microscope works in a similar principle. At the top here, is a high voltage electrical supply, and this emits electrons which are focused by magnets and accelerated as they go down this tube, and they hit a screen at the bottom there. And you take your sample of virus and you put it into this portal, which is this blue portal, which is halfway up the microscope. It goes in here and you bombard it with electrons. The electrons hit this screen here, a fluorescent screen, and when they hit the screen, they'll form light. This light can be detected on a photographic plate. That was the old way of doing it, as well as on the fluorescent screen. Nowadays, as you can see, you use a computer screen rather than the old method. Okay, so how do you actually get the viruses to show up in this beam of electrons? Well, when you do light microscopy, if any of you have ever looked at bacteria under a light microscope, or you've looked at things under a light microscope at school, you'll know that you use a stain. You use a stain which positively stains the thing that you're looking at different colours. That's called a positive stain because it's actively staining the things that you're looking at. With viruses, you use what's called a negative stain. A negative stain is something which stops the electrons, so it's impervious to the electrons. It's opaque to the electrons, and you embed the viruses in this opaque material. And what happens is that if you get the thickness right, the electrons won't go through the material where the stain is, but they will go through the material where the virus is. So when they go through the virus, they outline the structure of the virus. So you stain everything apart from the virus. You don't use the same stains that you'd use in light microscopy, obviously. You have to use stains that have a heavy metal in them, and usually that's uranium or tungsten. So what do you actually see? Here you see the negative stain, the black background. This is poliovirus, and you can see the outline of the virus, which is what you see in the electron microscope. This is a plant, and that, incidentally, that virus I just showed you had icosahedral symmetry. This is polyhedral symmetry. This one has helical symmetry. It's a plant virus called tobacco rattle virus. And you can see the structure here. It has a hole in the middle, and this helical structure contains the virus RNA in this case. Here's a coronavirus, and you can see it's showing up the envelope And that's why it's called a coronavirus, because this envelope has this sort of circle of spikes in it. And that's what gave it its name. When you see electron micrographs of coronaviruses, you'll often see they're coloured. That's completely artificial. The original electron micrograph is always grayscale. It's never coloured, because that's the way the electrons work on the fluorescent screen. Somebody, if you see a coloured electron micrograph, it's somebody who's put the colours in to make it look pretty. The original electron micrograph would look like this. So people have asked, where do viruses come from? Well, there are three ideas here. First of all, if any of you know anything about the way life was formed, in the origin of life, you'll know that one of the ideas is that RNA was formed before DNA, and that RNA life existed before DNA life. So the idea is that some viruses might be a very primitive form of life. There is evidence for this, because there are things called viroids, which are not really viruses, and they occur in plants, which consist only of RNA and don't have any protein. And they have the characteristics of what might be a very primitive form of life. The other idea that's been put forward is that these might be cellular organelles that have become renegade. Some structures within cells, like mitochondria and ribosomes, have their own nucleic acid. Mitochondria have DNA, ribosomes have RNA. These might have become renegade and have a different function and have started multiplying themselves. And so that may be another different idea for the origin of viruses. And then finally, there's this idea that they could be degenerate bacteria that have become adapted to an intracellular existence. And there's evidence for this as well, because there are bacteria called the Chlamydia and Rickettsia, which can only live inside cells. They're still bacteria, but they can only live inside cells. So they may be a sort of halfway stage. So which one of these is true? Well, it's very probable that they're all true for some viruses. And so viruses are not evolved from each other. They probably have at least these three different kinds of origin, and they probably originated in different ways. So unlike other living things, viruses are not necessarily evolved from other viruses. They could have a completely different origin. And there are these three ways that they could have originated. So this may be true, all three may be true for different viruses. Just to finish off this first session, I'll just show you some examples of common virus infections. Everybody probably has had these quite a few people have had these cold sores. These are caused by a DNA virus called herpes virus. And this is because the virus was latent in the nerve cells of your face and has suddenly erupted to form this cold sore. So this is herpes type 1. This is the other kind of herpes virus, type 2, which is a venereal infection and causes these fluid-filled vesicles, which is incidentally very infectious. There's some crossover now between the two, about 20% crossover. I'll leave you to figure out why that might be. Okay, so respiratory infections, I'll just finish off with this. Common cold is a very common kind of respiratory infection. The reason you can't get a vaccine against the cold is there are 100 odd different kinds of common cold virus, which are caused by rhinovirus. And this causes about something like 35% of common colds are caused by this virus, which is called rhinovirus. Coronaviruses, there are 4 different kinds of coronaviruses that we've all had probably before this one arose. And they cause about 20% of common colds. So we've all probably been infected with coronaviruses before. Incidentally, before somebody asked me the question, these don't give immunity against the virus we've got at the moment. And there are many other, well, several other different kinds of viruses that cause common cold. So common colds are caused by a whole gamut of different viruses. Flu, caused by 3 different serotypes, but the first 2 are the most common, and influenza A is the one that causes the periodic pandemics and epidemics. Believe it or not, there was a pandemic that swept the UK in about the last 3 or 4 years, caused by a DNA virus called adenovirus, and this disease called hacking cough. The reason people didn't pay it much attention was because it didn't kill many people, or it didn't kill any people. It causes a very irritating cough for 2 or 3 weeks. So that's another example of a pandemic which has occurred recently. Here's an adenovirus. It's a DNA virus which has an icosahedral structure. Just to finish off, the difference between colds and flu. Here you see some of the symptoms of flu. Here you see some of the symptoms of the common cold. The big difference between the 2 is in severity. If you've got flu, you really know it. If you've got cold, it can be various degrees of severity, which is not surprising, but usually it's mild. It's not surprising given the number of different viruses involved. Somebody says they have flu and they're still carrying out their normal life, they're going to work, etc. They haven't got flu at all, it's probably a cold. Flu, probably lays you up for 4 or 5 days, and it's quite a severe illness, and in fact can be lethal. Just to finish off, this is the last slide I've got in this session, bit of practical information. If you've got a sore throat, the most common reason to have a sore throat is a bacterial infection, streptococcal infection, which you see in the middle photo here. What you'll see, if you open your mouth and look in the mirror, you'll see these white spots or streaks on the back of your throat. That's the bacterial infection, the most common cause of a sore throat. And you can cure it, you don't need an antibiotic, usually an antiseptic lozenge will probably cure it. This is what a normal throat looks like. Hacking cough will give you this kind of red colour. No streaks, white streaks, just a redness. And that's what you get with coronavirus as well. You don't see these streaks. So if you think you've got coronavirus, it might actually be hacking cough. But if you've got this, the one in the middle, it's not coronavirus. And that's what you're most likely to have. All right, so just to finish off, I just mentioned that viruses, as well as being a thorough nuisance, can also be used. This is the second one here. The research that I was involved in was using viruses as gene therapy agents, for example, in cancer treatment. Viruses can also cause cancer. That's why we have a vaccine against cervical cancer, which is a vaccine against papillomavirus. So they're involved in cancer as well, but I haven't got time to go into that. I'll take some questions now and we'll continue with emerging infections a bit.
Thank you very much, Greg. I'm discovering that actually maybe science is understandable after all.
Well, I hope so.
So, as before, if you'd like to ask Greg a question, if you could indicate by hand, if you go against the bottom, you'll find an opportunity to put your hand up or to clap or something, but if you could indicate, and then Greg will endeavour to answer your question. We're running slightly behind, so, and you need to unmute yourself, of course, to ask the question. Running very slightly behind, so I suggest we take about 5 to 10 minutes worth of questions and then go into the second half. So are there any questions on on what Greg's had to say so far? No, all very clear.
Wow.
I
must have been clear.
I can't see any hands going up at all, Greg. Maybe people just need a cup of tea.
Could I ask something?
Please do.
Greg, so the thing about washing your hands, and washing surfaces, you know, that we're all doing because of coronavirus. So I think this was the first time I'd understood why soap and water is fine, because people tend to use really strong stuff on their hands and think that, you know, I mean, it's not as bad as Trump saying inject yourself with bleach, but there is a kind of sense that the stronger and nastier the stuff is that you use on your hands, the more protective it will be. But that's not true from what you said. Soap and water.
No, that's not true. Just ordinary soap and water, because this virus has got an envelope. As you know, soap will dissolve lipid. If you've got fat in something or something, it takes it away. So it'll get rid of the virus envelope. That inactivates the virus.
Right. So is it important that it's hot or warm water, or
no, not really.
No. More just using the soap and lathering up and doing it properly.
No, I don't. It's more comfortable, I suppose, for us if it's warm, but it doesn't have to be warm. No, it can be detergent as well. That's probably not so good for your hands.
Yeah.
Another virus, like you might. There's been a norovirus outbreaks in hospitals recently as well. If you go to a hospital where they're trying to guard against norovirus, you would not find soap and water. You'll find basically an alcohol sanitizer. The reason for that is because soap and water has very little effect on norovirus because it hasn't got an envelope.
Right.
We're lucky with this virus that it has.
We've got a couple more questions here, Greg. Maureen Wright would like to ask you a question. Maureen?
Hi, Greg. You've said that viruses have a preferred host, or that's what I gathered. And I was wondering about the transferring between species, given that coronavirus came from bats, I believe.
No, I'm going to say something about this in the second half. I maybe gave you the wrong impression then. They don't, they can very easily jump from one host to another. Flu does this, and coronavirus probably did. So when you say they're restricted to one host, it's usually something like mammals or birds or whatever. It doesn't have to be one species, and quite often it's not just one species. Some viruses just one species or a few species. Myxomatosis, which also is called a pandemic in myxomavirus in rabbits, mostly infects only rabbits and hares. It wouldn't infect anything else. So they vary a bit, but certainly they can go from one host to another, and it's actually quite common.
So I've got a couple more questions here, which I'll read out, which have come through on chat. This is from Emily Beattie. Is Synbis virus used in gene therapy? If so, why?
Somebody's been reading my papers. Yes, we were working on using Synbis and Semliki-Forest virus as gene therapy agents for things like multiple sclerosis and cancer. The idea there is that it's actually an RNA virus, not a DNA virus. So you put a gene into a therapeutic gene into the virus, and what happens is when it infects the host, it expresses the therapeutic gene as well as the virus genes. Of course, you can't allow the virus to multiply to its full extent in the host because it would kill the host probably. So you debilitate the virus as well. And we were using that, for example, for cancer treatment, and we were looking at multiple sclerosis as well, but that's in its very early stages at the moment. Greg,
you've got a bit faint. Could you come a bit closer to your microphone?
Yes, sorry.
Yeah, so I've got 4 more questions here. Emily says she hasn't read your papers.
Well, that must have been pure chance then.
She used to test for Synbis for diagnosis at Porton Down.
Oh right. Oh yes. Well, it's a mosquito transmitted virus, and you find it in tropical countries. I think it only causes a very mild disease in humans. That's really why we were using it, because it doesn't cause a serious disease in humans, although it can infect humans.
I've got 3 more questions. One relates to COVID-19 from Diana, and I'll suggest she asks that in the second half, because you're going to focus on COVID-19 in the second half. So the other two are, one's from Daniel Russ. He says, are wet markets a big threat to future pandemics?
It's something I'm going to talk about in the second half.
Okay. Come to that in the second half then, Greg.
I think, well, I'll just make one comment just to whet your appetite. It's quite possible that this wet market in Wuhan is a complete red herring. It may not have anything to do with the virus, actually. Be like Trump saying it escaped from a laboratory. I don't think either of those explanations is probably correct.
Right, okay, but you'll come back to that in the second half.
Yes.
And the other one that doesn't relate specifically to COVID-19 is, have masks been effective for any previous viruses? Will masks be an effective protective measure in general?
So you hit on quite a controversial subject there, masks. Well, it's complicated because masks aren't just of one type. They're really, I suppose, three different kinds of masks. There's the ones that are used, well, no, four actually. First of all, there's respirators, which people use when they're dealing with really dangerous viruses like Ebola or smallpox. Well, we don't do it with smallpox anymore, but dangerous viruses which take out all particles, and they're not disposable, but they have two disposable filters at the side, which are filters which take out all particles. Then there's the ones that are used medically, and there really are three kinds. One actually isn't used medically, it's the cloth mask. Then there's the surgical paper mask, and masks that are used to protect against viruses in a clinical situation. And these have a designated specification. So let's deal with the cloth masks first. They work to some extent, it seems, in stopping people being infectious to other people, in that if you cough and sneeze, they stop the big aerosol droplets, but they're not much use for stopping the small ones, and they don't protect the wearer really. So they're a bit useless. The paper masks are about protective to a certain extent, take out most of the big droplets, but are not totally protective. Now, the ones which are used in clinical situations are more than 95% protective, and they're sealed. They should be sealed round the edge. Wouldn't be much use on me because I have facial hair. Somebody who hasn't got facial hair, they would seal around the edge, so they'd be more protective. They have two different specifications. And it's usually written on them. They're called N95, which is the American specification, or the European specification would be FFP2 or FFP3. Those are the ones that are used in clinical situations in the PPE that people are talking about at the moment. Okay, so whether you should actually wear them in other than clinical situations is quite a controversial subject. Take quite a long time to cover that, probably a talk in itself.
Yeah, okay. I think probably enough on masks unless something crops up in the second half.
Nick, I've been noticing that Stephen Mason has had his hand raised for a long time to ask a question.
I have been looking, I apologise.
Oh, sorry.
But I didn't see it. So Stephen, do you want to ask a quick question? Then we'll move on to the second half. Just to say there are two questions people have asked on chat, which I think relate to the second half. So I'll make sure we come back to those.
This is really just to ask Greg if in the second half of his talk, he's intending to say anything at all about either the 1957-58 Asian flu pandemic,
yes,
which has been very little referred to, and or the 1968 Hong Kong flu pandemic.
Okay, I'm going to talk a little bit about flu virus and its pandemics in the second half.
Right, okay.
Great, okay. Would people, I'm going to suggest we just take a 2-minute break to give Greg a rest. So we'll start again at 8:28.
Okay, I'm just going to get a glass of water. I'm going to be
exactly. 2 minutes, 8:28.
Well, Nick, that was a lull at the start. I didn't think there were going to be any questions, and then they came rushing forth, didn't they?
No, but there's a few more here that I haven't asked you. And we've got 85 people in the room, so I think there will be a few questions.
When I talked to Dorset Humanists, there were a lot of questions about masks, actually. They seem to be very interested in masks. Not
Okay.
an expert subject, but...
Yeah. Maybe it's gone away a bit as an issue, I'm not sure.
Yes. There was one lady got very het up about the fact that she thought everybody ought to wear masks, but I can't say I've got any strong opinions one way or the other. But
some people are using the blue hand icon, but for some reason I'm not seeing it. So I'll ask people, and I can't see everybody anyway, so it's no good asking me to get them to raise their hand. What I'll do before we restart is to ask people to either use the blue hand icon or to type it in the chat where I can see it.
Is the hand icon on the reactions bit?
No, it's on the participants bit.
Okay, yeah.
But I'm not seeing it for some reason, Chrissy.
Oh, I see.
I'm going to ask people to use chat.
Right. Says we've got 86 people.
Indeed, 88 now.
Wow. Mm.
Right, let's get going again to make sure we finish at 9, everybody. So anybody who's just joined could please mute themselves, that'd be really helpful. We've got a couple of questions that were asked in the first half that Greg will pick up at the end of the second half. And for some reason, I'm not seeing the blue raise hand icon. So some people have been sending me a message on the chat, which is absolutely fine, and that works really well. So by all means, use the blue raise hand icon, but if I don't see you, then just send me a chat and I'll read out the question. So yeah, if everybody could mute again, and we'll go back to Greg for second half. Thanks, Greg.
okay, let's just bring that up again. Oh dear, I'm having trouble with I just have to go to the slide I got to.
Okay.
I tried to do that manually,
Yeah.
a quick way, but it seems to have gone to this. So
Scroll through.
just scroll through it. Yeah. Okay. So what I'm going to talk about now in the second half, what is this? It's, oh dear,
Yeah,
a bit late. So
a bit late, yeah.
I'm going to talk about what we can learn from viruses that have emerged, and I'm going to come to coronavirus at the end. But what I would say about coronavirus is that I retired a long time before this appeared, so I'm not really in the know anymore, but I'll give you a few thoughts about it anyway. But what I thought to begin with, I'll just talk about some other pandemics which have occurred, and somebody just mentioned then flu virus. I'm going to talk about that. But let's just look at what's happened in the past. Until quite recently, until about 1900, infectious diseases were a big problem. 40% of all children used to die before the age of 5, and this was usually due to infectious disease, and this was right up until 1900. A number of factors have caused this to tail off. One of the biggest ones was sanitation, and one thing made a huge difference, which was the chlorination of water, and introduction of vaccines also made a big difference. And here you can see that what people used to die from, and here you see the top 3 causes of death in 1900 were all infectious diseases. If you go to 1997, all the infectious diseases have disappeared apart from pneumonia and flu, which is way down the list, and HIV. So infectious diseases were a much bigger problem in the past. and they were caused by a lot of different infectious agents. You see here some of the bacteria and the viruses, things like smallpox, polio, measles, mumps, chickenpox, and these two viruses at the end, which cause diarrhea and still big problem in developing countries. Two viruses have been completely eradicated by the use of vaccines, and vaccines have been a big factor in reducing most kinds of virus infections, reducing the mortality, and maybe there will be with coronavirus as well. We don't know that yet. But two viruses have been eradicated by vaccines. Smallpox, which had a 20% lethality in the past, and many of the survivors were disfigured, but that's been eradicated by Jenner's original vaccine, actually. And the other one that's been eradicated is that virus of cattle, rinderpest virus, which was a big scourge in Africa and Asia, and in wildlife as well as cattle. So those have been eradicated by vaccines. Polio is nearly eradicated by vaccine, but it persists still in a small area of Pakistan and Afghanistan, which is controlled by the Taliban, and they don't like vaccination, so that's why it persists. So the threat's still posed. Well, new disease-causing agents, which we're seeing at the moment, one of them. And the other thing that's causing big problems in recent times is vaccine denial and hesitancy. And I'm going to, well, antibiotic resistance, that's a bacteria. As for bacteria, I won't cover that. So let me deal with a few of them. Let's deal with HIV, which is the pandemic which we've seen most recently, that's had high mortality before the present one. This first made an appearance in 1988, and you remember in gay people, it had high mortality to begin with, but now the number of people that are living with AIDS has gone up and is still going up. Number of new infections is decreasing, but not at a very high rate, but the number of deaths is also decreasing. And deaths now occur mostly in developing countries. The disease has mostly been controlled in developed countries through the use of antiviral drugs. The peculiar thing about HIV, the virus that causes AIDS, is that there's never been a vaccine developed. And the reason for this is that the virus is too variable, that one of the reasons anyway, even in a single individual, it shows huge variation, and that's why you can't immunize somebody against using a single vaccine against AIDS, but antiviral drugs have been successful there. Somebody asked about viruses coming from animals. This is the animal that HIV came from, chimpanzee, came from a virus called simian immunodeficiency virus, which infects chimpanzees and really doesn't have much of an effect on the chimpanzee. But how it came to get to humans is really unknown, but we know now for sure that it did come from chimpanzees. Ebola is another one. This caused an outbreak recently, which you probably all knew about 4 or 5 years ago in West Africa, in these countries here, Guinea, Sierra Leone and Liberia. And this has a very high mortality for a virus. It kills 40% of people that it infects. And we didn't have a huge number of deaths in terms of virus infection, killed about 11,000 people in that original outbreak. The reason why it was contained was because we understood how it was transmitted. And it's not actually very infectious, and the way it was being transmitted was through touch, through contact. And as soon as that was brought under control, we managed to keep the virus under control. Now, this is a success story in that a vaccine was developed very quickly, mostly by the Wellcome Trust putting a lot of money into it. And it was a novel vaccine, which was a genetically engineered vaccine. It was a cattle virus, and they put the genes that induced immunity against Ebola into the cattle virus. Cattle virus doesn't cause disease in humans, but it induces immunity. And if you put this gene into it, it induces immunity against Ebola virus. And that's been an effective vaccine. And what people, I think, haven't read too much about is that there's been another outbreak of Ebola in the Congo recently. And this has really been controlled by the use of this vaccine, which was developed by ring fencing the virus outbreak using the vaccine. There's another vaccine which has been developed. The first one was developed by Merck. The second one now that's being used at the moment is a slightly better one, which was produced by Johnson & Johnson and was based on a human adenovirus, like the virus that causes hacking cough, but by debilitating the adenovirus. This is of interest. It's an effective vaccine against Ebola, but it's of interest because at Oxford, the group in Oxford is using a similar method to try and develop a vaccine against coronavirus. So again, somebody very astutely asked about animals. We know as well that Ebola virus came from an animal. It came from a fruit bat and probably via an intermediate host. All these other animals can be infected by Ebola virus, and it might have been one of these which spread it to man eventually, but we know it came from fruit bats. And bats are quite a significant source of new viruses. We think originally rabies virus came from bats. Now I'll talk about flu virus. Somebody asked me about that. First thing I want to say about flu virus is that it's an enveloped virus, and it has two kinds of spikes, not just one. These, in this model here, they're shown as red and blue. Incidentally, flu virus, because it's an envelope, you can inactivate it in the same way as you can for coronavirus by washing your hands in soapy water. But it has two kinds of spikes, not just one. And these two kinds of spikes are what most of the immunity is directed against for flu virus. So what happens when you get a new flu virus strain appearing? Because what happens with flu is that you get pandemics in exactly the same way as you get in for other viruses, particularly coronavirus, but these occur at regular intervals. You get a new kind of influenza virus arises. People haven't got any pre-existing immunity to this new influenza virus. So it gets transmitted very easily throughout the whole human population and replaces the previous strain, which had induced immunity. So this occurs regularly with flu. So the formation of these pandemic strains which sweep the world, is caused by a process called antigenic shift. What happens here? Well, flu virus is an RNA virus, but it's peculiar because it's got 8 different segments to its RNA. The RNA isn't just in one piece, it's in 8 different pieces in the virus particle. And each of these code for a different protein. In particular, 2 of the segments code for the 2 spike proteins which induce immunity. So what happens in antigenic shift when you get the formation of a new flu strain? What happens is these 2 segments change completely, but all the other segments remain the same. So the virus now is able to get around the pre-existing immunity, but is able to replicate very well in human cells because the rest of the pieces of the genome are all still the same. Now, it's now known that these new pieces of RNA mostly come from birds. Birds get influenza very regularly. Influenza strains are very prevalent in birds, not just chickens, but all kinds of birds. But these strains don't infect humans. So how do the segments from bird strains get into human strains? Well, here another animal is responsible. This is the pig, because pigs could get infected with bird strains and they can be infected with human strains. If they get infected with both, then as a very rare event, you get the formation of this hybrid virus. And this is the one that causes the problems. This, funnily enough, nearly always occurs in China, believe it or not. Nobody really knows why that is, other than the fact that pigs are quite common in China and they often live in proximity to humans, but that's only a guess at the moment. So that's what happens when you get a completely new strain of influenza. This process has a technical name, it's called genetic reassortment, and it causes these regular pandemics of flu. Now, the question then is, can a virus like the coronavirus do this? Well, the short answer is yes, but it doesn't have a segmented genome. It has a genome that's all in one piece. But how can it do this? Well, it could do it because it's got a very large genome, the largest of any known RNA virus. And what this means is that the RNA pieces can break and rejoin very easily. So, and this actually has probably occurred when the coronavirus was formed in the first place. So coronavirus RNA can break and rejoin. So theoretically, it could do something very similar to what this flu strain is doing. Whether it will or not, that's a different question. All right, so that's not the only thing that flu can do, because we all know that every year the flu virus changes. We have to have a different vaccine every year because the flu strains from the previous year are not infecting us anymore. There's new strains have arisen. And this is a gradual process. These are the epidemics that occur in between the pandemics, and they occur every year. This is called antigenic drift, and what this is a process of mutation. The virus is mutating to change its two spike proteins so that it gets around pre-existing immunity. So this is why we have to have a new vaccine every year, and this is what actually occurs with HIV as well. Can this occur with coronavirus? Again, the answer is probably yes, because many different coronavirus has been sequenced now. Quite a lot of different strains have been sequenced, and there are many different strains, all of which are showing different sequences. Whether they differ in the immunity they induce or the disease they cause, we just don't know, but we know that it is capable of that. So these are the main flu pandemics that have occurred in the past. Somebody mentioned Asian flu. This is this one. This was the 1918 one that killed 50 million people, or this one here rather, that killed 50 million people. And this has occurred at regular intervals. This is the Hong Kong flu here. So this is when we're getting pandemics of flu. This is the H and the N just stands for the two spikes of influenza, and you can see they're given different numbers depending on the different kinds of spikes they've got. This was the one that killed so many people in 1918. Because people aren't immune to this anymore, it's made a reappearance. So this is circulating at the moment. Luckily, it's become attenuated. That's what the star and the PMD stand for. So it's not killing as many people now, not nearly as many, but it's still it's made a reappearance. And for the first time ever, we've got another strain circulating at the same time. So that's what happens with flu. Another analogy is this. This is a photograph taken in 1918 in the great flu pandemic that killed 50 million people. They were using warehouses as makeshift hospitals, putting very large numbers of beds into warehouses like this. So you can see a parallel with what's happening now with the Nightingale hospitals. Something similar is happening. So how can this occur in the future? Well, birds have a very large number of different kinds of hemagglutinin and neuraminidase that could be picked up. And 4 have made appearances in recent years and have rung alarm bells, but haven't actually spread. But we're waiting for a new one to appear. Just mentioned a couple of other viruses that cause problems. You'll remember Zika virus at the last Olympic Games. This caused microcephaly in newborn children, a kind of birth defect. It's a mosquito transmitted virus. You remember that was a problem at the time of the last Olympics, but seems to have disappeared now. Another one is chikungunya virus, which has caused problems in the Indian Ocean and in Asia. Again, a mosquito transmitted virus. Just a quick word about measles virus. Measles virus was once a very common disease. People think it's a mild childhood infection, but in fact, that's not really the case. In 2017, which is the last year I could find statistics on this, there were 110,000 deaths from measles virus in the world, and these are mostly children. In developed countries, 1 in every 1,000 measles cases gives encephalitis, which is brain damage. And even in developed countries, 2 out of every 1,000 children will die from measles infection. So it's not a mild disease. If you look, it's mostly been controlled by vaccines up until recently. You see here, when the first vaccine was introduced, the incidence dropped greatly. And then the MMR vaccine was introduced in 1988, went to near 0. We're lucky with measles because it's not got much antigenic variation. It doesn't vary too much. I was involved with this guy in 1998, believe it or not, Andrew Wakefield. He was the guy who caused the scare about autism and measles. The UK government set up a committee to look into his claims, and because I was working in Ireland, they wanted people from outside the UK on the committee, so I got put on the committee. I met this guy twice. He's a very charming guy, got the gift of the gab, as they say, or blarney, as they say in Ireland, but he caused enormous problems by creating one of the biggest frauds in medical history. Absolutely nothing to his claim that that measles vaccine is linked to autism, and that's been shown by 5 very big studies now. Trouble is that he caused, he was struck off the medical register for fraud, by the way, for creating that fraud. And this shows you what's happened with measles. It's at the moment got about 95% coverage, which isn't enough to give complete immunity, to give herd immunity. A lot of several outbreaks have now occurred of measles and in mumps because people aren't getting vaccinated. They think it causes autism, which is completely untrue. This is a bit out the Washington Post about an outbreak that occurred last September in New York among the Jewish Orthodox community. For some reason thought that vaccine was damaging their children. They stopped vaccinating their children, and a big outbreak of measles occurred. So now let's, in the last few minutes, get on to the current coronavirus. As I'm going to say straight away, I'm no expert on this because I'm not in the know anymore, but I'm going to give a few thoughts and people can pick them up in the discussion if you like. This is the designation of the virus, which is made by the International Committee for the Taxonomy of Viruses, and COVID-19, which you'll see in all the newspapers and news coverage. This is actually the name of the disease, which was designated by the WHO. So this is like the difference between HIV and AIDS. This is the virus and this is the disease. It's related to SARS and MERS, which were coronaviruses which emerged, caused quite a number of deaths, 800 in the case of SARS, a few more, about 2,000 or 3,000 in the case of MERS, and then disappeared again. We thought that was going to happen with this coronavirus, but of course we all know what did happen. Okay, so where did it come from? The Chinese are claimed, at least at the beginning, that it came from Wuhan meat market. Well, they've traced the earliest contacts now, and it seems a lot of them never went near Wuhan market. We do know that it's probably a hybrid between a bat virus, you probably derived from fruit bats, and a virus that infects pangolins, which are a kind of animal. I don't know whether you know what a pangolin is, but apparently it's the most traded kind of wildlife in Asia. So it's a hybrid virus. It's undergone this process of recombination, of breaking and rejoining its RNA. Now you can see here this very pretty electron micrograph. This isn't what came out of the electron microscope, of course, but that's just to remind you what it looks like. Peculiarity of this, I think we covered hand washing and face masks in the question. So I'll just point out, this is a unique kind of virus, the way it replicates its RNA through what is called a nested set. So no other virus replicates like this. And it's got the largest genome of any known RNA virus. We all know the basics of the disease it causes. It's older people that are most susceptible to or to disease and who are subjected to the greatest mortality. Children, funnily enough, as you can see here, are very little affected by coronavirus, and this is quite different to a lot of other viruses. And as we know, people that have underlying health conditions are also susceptible. And the peculiarity that no one can really explain about this virus is that there's a lot of ideas, but we don't know what the answer to this is, that twice as many men as women die from the virus. This is a peculiarity which I'm going to point out because I think it's quite an important peculiarity. If you look at the way coronavirus cases are counted, they're usually counted according to tests that are done of people who've shown symptoms of the disease. This was done early on in China, and it was done in the UK as well. So you've probably, this is called the iceberg effect. You've probably got a very large number of cases of people who have only had mild disease or perhaps even no disease at all, and are not showing up in these statistics. Just how many these are is another question. But this was a way they illustrated this in the Financial Times, and could be illustrated by what's happened in Germany and the UK as regards testing. In Germany, they got a death rate of 1.3%. In the UK, in the early testing anyway, it was 8.7%. And this seems to have been mainly because of the way the testing was done and the way the deaths were counted. In the UK, the only people that were tested, at least at the beginning, and this I gather has changed now, but the only people who were tested were the ones with either had died or had severe disease. So you've got a high death rate. In Germany, they did a track and trace testing. So they tested a lot of people that had only mild or moderate illness. And that's the reason they got a lower death rate. But they're still not testing people with no symptoms. Iceland is the only country who did this early on. They allowed anybody to, and again, this wasn't random. It still wasn't random. What they did was test anybody who turned up who wanted to have who wanted a test. And they were usually people who'd been in contact, but not always, usually people who'd been in contact with somebody who was known to have the disease. They ended up testing 10% of their total population. Not a very big population in Iceland, it's only 360,000, but they tended, so this was nearer to random. Lo and behold, they get an even lower death rate. And even this isn't random. So what can you conclude from this? Well, it looks as if the actual, and I'm not belittling here, I must say straight away that I'm not belittling the trauma and the human tragedy of people who've died from coronavirus. I really, that is really very serious and tragic. But what I'm talking about here are statistics. I think, to be honest, this virus is much less lethal than we think. I read a report in a journal, just a paragraph today, that Germany has started serological testing, a different kind of test to the PCR test that's done to detect the virus genome and detects anybody who's had the virus. And they've done this at random, and they're getting 10 times as many people showing no symptoms as show symptoms. So it looks like, I'm putting forward the idea anyway, that there's good news and bad news here. The good news is that the mortality is much lower than we think. But the bad news is that the virus might have spread much further than we think as well.
Let me check how many more slides you've got.
I'm nearly finished now.
Great. Okay.
Got to the, yes, it's 9 o'clock. All right, I just wanted to make a quick comparison to what's happened in the past. With flu, you get an average of 17,000 people dying a year. And in one pandemic year, the latest one, it was 28,000. So we're actually only on a par with this now in terms of coronavirus deaths. Flu is actually higher worldwide than coronavirus deaths have been so far. A norovirus kills 200,000 people a year, and these are mostly children actually, not adults. In 1918, the flu virus killed 50 million people worldwide. And in the UK, it was well over 200,000 when the population was only 42 million. So you can see how these compare. All right, so you can see here, I'll skip that slide because it's not very important. I just wanted to make the point that we're the 5th highest mortality of any country in the world. People have wondered why that was, and the last slide here might give some insight as to why, and I'll finish on this. Some countries have been very successful in containing coronavirus. These are just 5 of them: Taiwan, Hong Kong, South Korea, Finland and Germany. Less successful are these countries here. So what's the difference between the two? Well, there's been all kinds of things put forward: wearing of face masks, stringency of the lockdown. Even, believe it or not, I saw an article on CNN to say that these countries all had women leaders, and that was the reason. South Korea, of course, doesn't have a woman leader, and Belgium, which is the worst country of all in terms of coronavirus deaths does have a woman leader. But anyway, that's an irrelevancy. I'll tell you what I think it is. I think it's the fact that they all went in for tracking and tracing very early on, and we didn't do that here, and neither did any of these other countries. But I think that's going to be important when we start coming out of the lockdown. Sweden is an interesting one because they've had no lockdown at all, only voluntary. All their schools and shops and other institutions have stayed open. Children are still going to school, and yet their mortality rate isn't as high as ours, although it's higher than other Scandinavian countries. So what's happening now? Well, various antiviral drugs are being developed. I've put some of them here. Antibodies targeting the virus. They're talking about using convalescent sera now. Drugs which target the viral RNA polymerase. The only one that's shown any kind of effect so far is this one, remdesivir. That doesn't really affect mortality, just the speed of recovery. And there, as I've said earlier on, there are various genetically engineered vaccines under development. So where are we with this? Well, it's easy to try and counter the anti-vaccine pseudoscience with measles, etc. But with the coronavirus, we should have tackled it earlier on. We should have tackled it with a proper plan to tackle initiation of a pandemic early on, which they did in those countries that I mentioned. We haven't done that, but tracking and testing, tracking and tracing is obviously going to be important as we go out of this lockdown that we're in now. Okay, well, I'll end there. Thank you for listening to me, and I hope we could take a few more questions. Right. Sorry we're running late, Nick. I'm sorry about that.
Don't worry, that overran a bit in the first half. If you don't mind holding on for 10 minutes or something, Greg, then we've got some questions